In this video lesson, we'll have a detailed look at the digestive system and the processes involved in taking a steak or an apple or any other item of food or drink and breaking it down into the nutrients that can be absorbed into the bloodstream and used by our body for energy or to build new cells and cell components. In particular, we'll look at the organs involved in digestion, including the mouth, the esophagus, stomach, the small intestine, which is made up of the duodenum, ileum, and jejunum, the colon or large intestine, and then finally the rectum and the anus. Digestion starts in the mouth with the action of the teeth, jaws, and the muscles lining the mouth working together to break food down into pieces small enough to swallow. Chewing also gives food a larger surface area for digestive enzymes to access in later steps of digestion. Foods high in fibre, such as an apple or a piece of celery or whole grain toast, stimulate chewing and saliva secretion. Chewing, therefore, is a type of mechanical digestion as it involves movement to break food down into smaller particles. The second main function of digestion in the mouth involves saliva, which is secreted from salivary glands located around the mouth and jaw area, as shown in the image at right. Saliva moistens the food to allow easier passage through the esophagus, for example, and down into the gastrointestinal tract. Saliva also contains the enzyme amylase, which breaks down starch into simple sugars, particularly maltose. This can also be seen in the image at the bottom right, showing large carbohydrate molecules being broken down into simple sugars. The action of saliva can therefore be considered an example of chemical or enzymatic digestion. Did you know? The sight, smell, taste, or even the thought of food can increase the amount of saliva secreted. The next step of digestion involves swallowing, where the tongue and cheeks push the food into the teeth and shape food into a bolus or a round ball, which is able to be swallowed by the pharynx and passed down into the esophagus. The esophagus transfers the bolus from the mouth to the stomach in a few seconds. The bolus is pushed along by a process known as peristalsis, a wave of contractions and relaxations of muscles lining the esophagus. This can be seen in the image at right, with the arrows indicating areas of contraction and relaxation, progressively moving the ball of food down the tube. Did you know? People do not have conscious control over the muscles in the esophagus. Even if you're upside down, food will be naturally passed on to the stomach. This is an example of involuntary muscles. Next, we move to the stomach. And first, we're going to focus on mechanical digestion, which is the digestion involving movement of muscles. The stomach is an expandable sac that can hold up to 1.5 litres of food and liquid. Muscles in the stomach wall churn the bolus for up to 3 to 4 hours. As you can see in the picture at right, there are several layers of muscle lining the stomach, making it very strong. Once inside the stomach, the bolus is mixed with hydrochloric acid to kill any pathogenic bacteria present and provide the right pH environment for enzymes for chemical digestion. Now we'll have a look at chemical digestion. Food is mixed with gastric juices in the stomach, including hydrochloric acid, as mentioned, and enzymes. The bolus becomes a creamy mixture known as chyme, a beautiful green colour shown in the top right image. The enzyme pepsin starts to break down protein to form peptides, small chains of amino acids. Note how the name peptide and pepsin relate to each other. You can see in the image at right a large protein molecule being digested into single amino acid molecules. Then, the pyloric sphincter, a ring of muscles separating the stomach and the duodenum, opens and the chyme is released gradually into the duodenum. The duodenum is part of the small intestine, a tube which measures about 6 meters in length if fully extended. Obviously, as you can see in the picture, it is tucked away to fit inside your stomach cavity. The small intestine itself is divided into 3 sections: the duodenum, the jejunum, and the ileum. We'll focus on the duodenum first. Once inside the duodenum, bile salts from the gallbladder are mixed with fats to emulsify large fat droplets into smaller particles that can dissolve in the chyme. In the diagram, imagine the green circles are bile salt molecules, whereas the lipids are the yellow. Bile salts are able to attract to both fat molecules and water molecules, and hence, when they surround individual lipid molecules, they make these molecules able to be dissolved into the surrounding watery mixture of the chyme. This is the first step in lipid digestion. Next, pancreatic juices from the pancreas containing more enzymes are added to further digest carbohydrates, proteins, and fats. Pancreatic juices contain sodium bicarbonate, which is alkaline or basic, to neutralize the hydrochloric acid from the chyme. Pancreatic juices also contain digestive enzymes to break down nutrients, as follows. Polypeptides are the peptide chains that were broken down from proteins in the stomach. These are further broken down into individual amino acids by the enzymes trypsin and chymotrypsin. Starch, on the other hand, a type of carbohydrate, is broken down again into maltose by pancreatic amylase. Fat, or lipids, are broken down into fatty acids and glycerols by pancreatic lipase. Next, we move to the ileum, the second component of the small intestine. The inner surface of the small intestine is folded into finger-like structures called villi, which can be seen in the images to the right. Villi greatly increase the surface area available for the absorption of nutrients. In fact, the villi in your small intestine have a surface area of about 200 meters squared, which is equivalent to the size of a tennis court. So imagine emptying the contents of your stomach over the size of a tennis court and seeing how much of that could be absorbed and at what rate. Examples of digestion which occur in the walls of the small intestine or along the villi are peptides being broken down into amino acids by the enzyme protease, maltose being broken down into glucose by the enzyme maltase, sucrose being broken down into glucose and fructose by the enzyme sucrase, and lactose being broken down into glucose and galactose. Can you guess the name of the enzyme this time? That's right, lactase. There is clearly a relationship between the enzyme name and the substrate or the starting molecule. So protease and proteins, maltase and maltose, sucrase, sucrose, and lactase and lactose. It makes it a little bit easier to remember what's going on here. So that's the end of the digestive stage, where nutrients are broken down into their smaller components. Now comes absorption of nutrients. So water, alcohol, sugars and mainly glucose, minerals, vitamins, amino acids from proteins, fatty acids and glycerol from fat are all absorbed by the villi lining the walls of the small intestine or ileum. Once in the bloodstream, the nutrients are transported to the liver. The liver then converts the nutrients into substances that can be used for energy and growth. Undigested substances pass into the large intestine or the colon. The large intestine is a tube measuring just over 1 meter in length, and as you can see from the image, comparing to the small intestine, is a wider but shorter tube. Its main function is to reabsorb water into the bloodstream. Time spent in the colon is also the last opportunity to extract nutrients from dietary fibre, which has remained undigested as it's passed through the stomach and the small intestine. Bacteria in the colon can ferment dietary fibre and therefore produce fatty acids and gas. Some bacteria also produce a beneficial nutrient, vitamin K. Water and any remaining minerals are absorbed, leaving only waste behind. A little more on gut bacteria. The gut contains between 400 and 500 different species of bacteria, some of which are pathogenic and some which are beneficial. They're measured in colony-forming units per millilitres. Probiotic-containing foods, for example yogurt, kefir, and kimchi, and probiotic supplements can boost the population of beneficial bacteria and hence allow us to be more efficient in our digestion in our colon. After passing through the colon, the watery residues that remain move into the rectum, where they are stored, before being excreted as feces through the anus. This whole process can take between 12 and up to or more than 24 hours to pass all the way through. This time can be reduced if the diet is high in fibre. In the image at right, you can see the relationship of the colon, the rectum, and the anus. There are obviously a number of other semi-structures that are labelled here. You don't need to worry about those. The main ones are the colon, rectum, and anus. As a bit of an aside, the Bristol stool chart is a bit of a giggle really, showing different types of stools that are possible. So what type are you? To make the most of what you have just learnt, you should now go on to describe the digestion of one of the following meals, either a meal of fish and chips, salad, or a hamburger. To get started, list the main nutrients present and then follow each nutrient through the digestive tract.